It will take some time to be up, but it's uploading.
Yeah, great.
So, yep, the cilia is a projection like this on the cell. If you take a cross section of it like this, you will see something like this. So what is this?
Doc, your voice is breaking. Doc, hello.
Hello, can you hear me? Hello?
Doc, sorry, your voice is breaking. Doc, I couldn't hear you.
Still? Is it better now?
Yeah, it's better, Doc.
Okay, good. So, yeah, we're just speaking about the cilia structure. This is the cross
Mm-hmm.
section of a cilia, and inside of it, it has microtubules. It is, well, it's not important, but they do mention it in the literature that you have this shape, okay? So it's circular. So there is 1, 2, 3, 4, 5, 6, 7, 8, and 9 doublet. What does it mean, doublet? It means you have two of them, yeah? Can you see? An alpha
Uh-huh.
tubulin and a beta tubulin, yeah?
Yeah, Doc.
Okay. So 9 doublet and two microtubules as a singulate, like one on its own in the middle. So does this one make sense? So it is 9, they write it like this. 9 multiplied by 2. Okay?
Okay, Doc.
So
9 doublets and 2
I'm sorry, they write it,
singular arrangements of
yeah.
microtubules. Okay.
So they write it 9 multiplied by 2 plus 2 multiplied by 1. Yes, that's how they write it exactly. So what does this mean? 9 multiplied by 2, that the 9 are doublet, while the 2 are singulate. Okay?
Okay, only the circles you're talking about, right, Doc?
Yes. The circles mean tubulin basically, yeah?
Yeah, Doc.
Okay, so we have something called the basal body. This basal body, well, the question is, okay, I have the cilia, where it comes from? I mean, where it begins. Well, if we have the cilia like this, we are taking a cross section here. What if we take a cross section here, like near the base? Yeah?
Mm-hmm.
So this is the basal body. Base of cilium below cell membrane. It consists of 9 microtubule. Now it's a triplet. So can you see? 1, 2, 3. 1, 2, 3. So we say 9 multiplied by 3 plus 0 multiplied by 0. Okay?
So there's nothing in middle, right, Doc? Only in the periphery we have.
Exactly. Make sense?
Yeah, Doc.
Okay.
The triplet.
So that's the basal body. Okay, so next we have something called dynein. You know what is dynein? You tell me what is dynein.
Dynein is from positive to negative, Doc. It goes retrograde for the retrograde movement, Doc.
Okay, so the dynein, just a second. Yes, I hope it will be a good one. So I just want to show you like the movement of the thingy. So this is a spermatozoa, for example. Can you see the 9 and the 2?
Yes, Doc.
Yeah, so it's like this, 9 and 2. And next thing, there is this dynein arms. Can you see the dynein arms?
Yes, Doc.
These ones. Yep. So basically they help to move the tubule. So you think about it. If the movement is going to be like this, it's going to make it rotate, correct?
Okay.
Yeah, so this is, see? Now look what happens. It's going to swing. Yes, like this. Yeah? So there is going to be a movement leading to up and down, up and down. Yes?
Okay, Doc.
Okay. So this is all because of the dynein arm. Okay, so they write here Gap junction Yes, dynein, it's an ATPase. Also, kinesin is an ATPase that links peripheral 9 doublets and cause bending of the cilium by differential sliding of the doublets. Okay, you saw how the sliding mechanism go, yeah?
Yes, Doc.
There is something called gap junction. Where did we see gap junction before? I feel like I saw it before. Where did we see it before?
Not in here, Doc. Not
It
in
was in
biochem. I don't
cardio,
think so.
but what was it doing in cardio?
Hello?
Yeah, it was in cardiology.
Gap junction, maybe for dimples, Doc. I say no dimples.
Yes. So they used to propagate the impulse from cell A to cell B. Yes?
Yes, Doc.
Okay. So, gap junction enables coordinated ciliary movement, which means it allows the cilia. So we have many cilia. The cilia can speak with each other and make their movement happens at the same time. Okay?
Yes, Doc.
Good. Cartagena syndrome. Autosomal recessive dynein arm defect. Can you tell me what is cytosine versus what is dextrocardia? Why patient with Cartagena cannot have children? Why do they have bronchiectasis?
Basically, they have a problem with the dynein arm, right, Doc? The dynein arm is used in the swimming of the sperm, right? So that's why they have immotility in the sperm, and that's why they are infertile. And also, the cilia is made up of dynein arms, no? And it's not able to clear the mucosa.
Mm-hmm.
So that's why they have bronchospasms.
Yeah, their mucociliary apparatus is pretty much bad. Yeah, because no cilia, so they cannot clear it out. They get it. Okay.
So, Doc, this cilia structure is present in dynein and kinin, Doc?
Cilia structure? I did not get this question.
So, what is the dynein arm? Is it a part of cilia structure, Doc? Dynein arm?
Yeah. So if you take a look at this microtubule, can you see this little thing here?
Okay. What about the kinin arm, Doc?
Kinesin, you mean?
Yeah, kinesin.
It should be also existing here, but they don't draw it.
Okay. So cilia has dynein arms.
Yep.
For it to move you showed me, right? It moves the actin arm. Right, Doc?
Yeah, it leads to this sliding movement.
Yeah.
Okay. So
is it sliding movement of the actin arm, Doc? What is it moving?
Well, if you imagine this sheath, outer sheath, and you have this thing in the inside, yeah? So the sliding movement makes the upper sheath go back and forth, which lead to a movement. As I showed you in the
understand,
animation.
Doctor, but the upper sheath, is it the actin or the actin filament?
I'm not sure. I'm not sure what it is made out of.
Okay.
I'm not sure. Well, here we have Cartagena syndrome. Can you tell me why they are infertile?
Because of cilia, Doc. The dynein arm, there's no movement. It's required for the swimming, right, Doc? For motility.
Yes. Okay. Can the lady be pregnant with the IVF?
Yes, Doc. 100%.
Okay. And what about... Yes, finally. Okay. What about the next one, which is... Let's see. Yes, they write immotile cilia, dysfunctional ciliated epithelium. So finding will be developmental abnormalities due to poor migration and orientation because of the dynein arm. So they get situs inversus. So their spleen will be where? Where the spleen will be in situs inversus?
It will be on the right side, Doc.
Correct. Liver?
Liver should be on the left side, Doc.
Kidney?
Kidney?
Actually, it's a real
we have
question.
2 kidneys, right, Doc?
Yeah, but look, normally the kidneys are going to start facing it like this. They are going to flip. Yeah, they will stay the same, but they are going to flip.
Okay.
Yeah. What about the heart?
Heart on the right side, though.
What is the point of maximal impulse?
I don't understand. What do you mean by that? Maximal impulse?
Point of maximal impulse, it means where is the apical impulse?
Oh, probably sternum.
No, man. Remember when we spoke about the apical impulse in a normal human being? It was on the fifth intercostal space, 2.5 centimeter left from.
Got it. But
Yeah,
that's normal. You're asking about dextrocardia, right?
absolutely. It's the exact same thing, but on the right, not on the left. So it's here. See? It will be like
Oh,
here.
so even the orientation, the axis will be like that, though, same?
100%.
Okay.
Yep.
Okay.
Yep.
Okay, though. I thought the whole heart is going to move to the left, exactly the same. No.
Exactly the same. So it's just a flip.
Okay. Mirror image. Okay.
Yep. So they write here, cytosine versus and hearing loss. Why hearing loss? Because our Caustian tube does have cilia in it. Okay. So recurrent infection can occur in the patient. Why? Their mucociliary apparatus is gone. So they can get recurrent sinusitis and ear infection. Correct?
Got it.
Also bronchiectasis. Why the bronchiectasis? Because of an impaired clearance of the debris and the pathogens, so they get the bronchiectasis. Also infertility, which increases the risk of ectopic pregnancy due to dysfunctional fallopian tube cilia. Remember, in the fallopian tube of a lady, there is this cilia, and basically, in the fallopian tube, this thing is super serious. Why? Because in the fallopian tube, they cannot propagate and move the zygote forward. You know what is a zygote?
Yes, though.
So they cannot move it forward.
It's not able to take it from the ovary, from the fallopian tube into the uterus.
Yes, so it's stuck there.
Got it.
And that increased risk of ectopics. And
yeah,
in man, it's immobile sperm. Okay,
got it.
The lab finding, which I was looking for, it's decreased nasal nitric oxide level. Each time I teach this concept, the same question. Please ask it.
I don't know, though. Why?
Why nasal nitric oxide is even related to dysfunctional cilia? Yeah? Well,
yeah.
Here it goes. Okay, let me show you. If you do have cilia, you will have nitric oxide produced by them. Okay? If they are working.
Okay.
If you do not have cilia, you will not have any nitric oxide produced by them. Okay? If you have, well, how do we test even for that? We bring the patient, we literally put something inside of his nose. Okay? And after that, you make him inhale some nitric oxide, and inside of the cell, a process is going to happen to convert that nitric oxide where? Inside of the cilia into something else. Okay? So that's something else, which you don't have to know. It's a reduced form of nitric oxide, is going to be detected. Okay?
Mm-hmm.
So why do we put the mouthpiece? Because otherwise he's going to suffocate. You need to keep a hole open, you know?
Okay.
So this is the nitric oxide test. Keep in mind that the results are demonstrated on something like this, on a graph. Okay?
Okay.
So yeah, this is the nitric oxide test. Basically, just check the function of the cilia.
Got it.
Good. So this is cartilaginar, this is the base of the cilia, and this is the cilia itself. Sodium potassium bump. Can you tell me where did we hear about it before?
Sodium potassium pump, we see it in the cardiomyocyte, doc, where Jobson works on it. Right? Mm-hmm. Any problem with that, indirectly it will block the sodium calcium exchanger, causing more contraction. Calcium induced calcium contraction.
Yes.
And we also saw sodium, potassium, and 2-chloride pump in the loops, the ascending loop of Henle. And sodium potassium ATPase pump we also see in the collecting tubule though. Aldosterone works on it.
That's correct. Good. Well, one more question. What is thyroid hormone going to do for this pump?
Steroid hormone.
No. Thyroid. As in thyroxine and 3-iodotropin.
Thyroid. Okay. Thyroid, what would in ATPase pump? Probably stimulates it though.
Okay, but actually, they do. Actually, that's true. But I want you to know it in a different way. They increase the presentation. So let's imagine you only had 2. They will put more. Okay? Let me explain. So the thyroid hormone is an intracellular hormone, correct? So it will go to the nucleus, it will bind to its receptor, and it is going to lead to overexpression of the sodium-potassium pump. Okay?
Yeah.
Overexpression of the sodium-potassium ATPase is going to make your body consume more or less ATP.
More ATP.
So that is going to do what to your metabolism?
Increase.
Yes. So in cases of hyperthyroidism, your metabolism is going to be really high, which makes you really, really, really thin.
Yeah.
Make sense?
Yeah, Doc.
Can you repeat this concept, please?
So basically, thyroid hormone is an intracellular hormone, and whenever we have increased thyroid hormone, there will be overexpression of the ATP pump, which is sodium potassium pump. And when we have increased sodium potassium pump, we have increased metabolism, and hence the weight loss.
Done. Okay, hypothyroidism now.
Weight gain, Doc, because less metabolism.
Done.
Because there's not much ATP pumps.
Yes, so no consumption of the ATP. Thus, we will have formation of a lot of fat. Make sense?
Got it. Yeah.
Okay, good. So actually, most of the time it takes me a difficult time to explain this concept, but you made it easy. Good, thank you. So, sodium potassium ATPase is located in the plasma membrane with an ATP site on the cytosolic side. Well, that's logical because the ATP is made inside of the cell. Yeah?
No, plasma membrane is where?
This.
Plasma membrane. What is plasma membrane? Like regular membrane, cell membrane? No?
Yes, it's a phospholipid bilayer.
Okay.
Yep.
So cell membrane is also called as plasma membrane.
Absolutely. Yes.
Or is it because it's facing the blood? No, not like that.
No. Plasma membrane is cell membrane, our cell membrane.
Okay.
So for each ATP you consume, well, think about it before we speak about it. Normally, which substance do you have more inside of the cell?
Inside of the cell is potassium, outside is sodium.
Okay. So to move sodium to the outside, will you need energy?
Yes, Doc, ATP. Yeah.
Why? Because you are pushing against the gradient. Yes?
Yes, Doc.
Okay, to move potassium inside?
We need ATP.
Yes, so look what they write. For each ATP consumed, 2 potassium are going into the cell, and that happens during the pump when it gets dephosphorylated, and 3 sodium goes out of the cell. And this happens when the pump is phosphorylated. Okay?
Okay, Doc.
So, cardiac glycoside, like aminoglycoside, the glycoside, I mean like digoxin and digatoxin. Digatoxin is not used anymore. Why? Because its therapeutic window is so narrow, was impossible to control it. Digoxin also have a really narrow therapeutic index, but still better than digatoxin. Digatoxin, however, is still used in the labs. So it directly inhibits the sodium potassium ATPase.
Got this. This I know from cardiology. So.
Yeah, it's the same exact thing.
Got
more
it.
calcium, more cardiac contractility. Nice.
Got it.
Okay, let's go to the collagen.
Okay.
So we have many types of collagen in our body. The important thing to know that their location. Remember what they say. So they write collagen, most abundant protein in the human body, extensively modified by post-translational modification. Can you give me examples of post-translational modification, by the way?
The cutting of the N-terminals,
And C-terminal, what else?
C-terminal, and capping and putting the tail on.
No, man. We are speaking about the protein. There is no tail on a protein. Tail and the cap
okay. So what were you
on
asking me?
mRNA.
What was the question?
Post-translation modification. Examples. You said remove N-terminal, remove C-terminal. That is correct. What else? There was something about biochemical procedures. If you remember, glycosylation, oxidation, all of this stuff, yeah? So, okay, so they write, type 1 is in the skeleton. Type 2 for the cartilage, 3 for the arteries, and 4 for the basement membrane. To be totally fair with you, this is the most important ones. So type 1, most common in the bone. Bone has 1 in it, yes?
Okay.
I mean, it has ONE, which is 1. So bone has 1, so it's type 1. Made by the osteoblast. And it exists also in the skin, tendon, dentine of the teeth, fascia, this is important, the cornea, and late wound repair. I want you to notice that early wound repair will be type 3, late wound repair will be type 1, which means that type 3 will be converted to type 1, okay?
Okay.
So they write, the type 1 collagen is going to be decreased in osteogenesis imperfecta, and logically, the areas that will be most likely to be harmed will be the dentine and the bone, yes?
Okay.
Okay, let's speak about the type 2, cartilage. So it's the cartilage, including the hyaline and the vitreous body and the nucleus pulposus. Tell me, what do you know about the nucleus pulposus?
It causes sciatica.
No, man. Oh, yeah, if it herniates. Yes. What else? Where it comes from?
It is from a neural crest or something.
Why you all guys say neuronal crest? No, it's not.
Neural tube, sorry.
Okay, so no, it's not from neuronal tube, it's from the notochord, yeah? And the notochord used to produce one substance related to some cartoon.
Yeah, the notochord has the Sonic Hedgehog gene.
Yes, what was the function of Sonic Hedgehog?
To fuse the brain development?
To make the neuronal tube, yeah?
Okay.
To convert the ectoderm into neuronal tube. So
Sonic Hedgehog, okay.
Again?
Sonic Hedgehog, right?
Yes, Sonic Hedgehog. You know, like the cartoon, I don't know if you watch it. So,
yeah.
Nucleus pulposus, it's in the middle of the vertebral disc. Yes?
Yeah.
Okay, good. So the type.
So what does it do, Doc? Sonic Hedgehog, what does it do?
It leads to conversion of the ectoderm into a neuronal tube.
Okay, Doc.
Okay. So the type 3. Type 3 makes something called reticulin, which is located in the skin, and the most important thing, it makes the blood vessel. Why it's the most important thing? Because it's associated with pathology. So it makes the blood vessel, the uterus, and the fetal tissue, and early wound repair. Remember, early wound repair is a 3, and after that 3 is converted into 1. So type 3 of Ehlers-Danlos syndrome. So this is collagen type 3. There is 2 types of Ehlers-Danlos syndrome, a disease we'll speak about today. Ehlers-Danlos syndrome, vascular type and musculoskeletal type. The vascular type, it's a defect in collagen type 3. Okay?
Okay.
Collagen type 3. The musculoskeletal type is collagen number 5. You are like, but there is no number 5 here. Yes, there is no number 5 here, but it will be existing there. Okay?
Okay.
The next thing is type 4. Oh, wait a second. Ehlers-Danlos syndrome, the vascular type. These patients, yes, they are going to have all of this aortic aneurysms, dissection, stuff like that. But they can ask you, what is the danger in a pregnant lady? The answer is a ruptured uterus. Yeah? Because their uterus is poorly developed and it might rupture. Okay, so the type 4 is in the basement membrane. Do you remember what I told you about type 4 before?
Type 4 is in the basement membrane, Doc.
Yeah, and it had like collagen type 4. And I told you the cancer cells, when it wants to destroy it, it produces what? Produces collagenase, which is a type of metalloprotease, yeah?
Yeah, Doc.
Okay, so it's in the basement membrane and also in the lens. Do you remember Albert syndrome?
Yeah, Doc. Alports.
Yeah, do you remember the lenticunos?
No, Doc.
Lenticunos.
Lenticulos.
This thing. It was in Albert syndrome. They had a defect in type 4 collagen, and thus the lens is made out of type 4 collagen. Yes? So its structure is going to become more weak, and it will kind of herniate. Yeah?
Okay, Doc.
Make sense?
I'm getting confused with retina and cornea. Lens, I mean. Okay. So type 1 is cornea, type 4 is lens. Okay.
Type 1 is cornea, type 4 is lens. Yes. Okay. If you forget about it, lens is 4 letters. Okay?
Lens is 4 letters. Okay, Doc.
Okay. So type 4. So under the floor, type 4, basement membrane. Defect in Albert syndrome. Can you tell me what else is problematic in Albert syndrome?
Can see, can't be, can't hear a B. So hearing problem, seeing, eyesight because of the lens, and kidney problem, nephritic syndrome.
Nephritic or nephrotic?
1 second, let me. Alfred Priam, right? Nephritic, though.
Okay. Well, which type of cast they will have?
Nephritic will have RBC cast.
Good. What is the difference between Albert syndrome and Goodpasture syndrome?
Goodpasture is only kidney and lung, dog. This one has even hearing, right?
Yep. And it's due to what?
Goodpasture is because of basement membrane, dog. This one is also basement membrane.
Yeah, both of them is basement membrane, but what is the difference? The pathogenesis. Well, Albert was a defect in the collagen itself, but Goodpasture was an absence of the Goodpasture was formation of antibody against the basement membrane. Yeah. So antibody mediated versus an actual deficiency of the collagen. Yes.
Yeah.
Okay. Good. Are you fine with collagen so far?
Yes, doctor. Where do we see Alport syndrome in the book, though? It's not in.
We saw it. We saw it.
Renal, right?
We saw it in renal under nephritic syndrome.
Yeah, but I mean pathogenesis also they gave it, doc. Let me just check.
I think we spoke about it.
Yes. So Alports, Rhodesian type 4 collagen, okay. And the pastures is got it.
Okay, good. So collagen synthesis and structure. Let's speak a little bit about the structure of collagen. How do we synthesize collagen? How do we do the post-translational modification of collagen? First of
So,
all,
Doc, in this one, Doc, the metalloproteinases. What was it about the metalloproteinase?
it helped, like the cancer cells, for example, help to destroy the collagen type 4 by producing metalloprotease.
The cancer cells. Okay.
Yep.
And anything to do with early to late wound repair, Doc? Metalloproteinase does it?
Yes. So when you want to convert type 3 collagen into type 1 collagen, you must remove the type 3. Yeah? How to remove type 3? Well, you need metalloprotease.
Okay, Doc. Where do we learn this, Doc? About
Pathology, second chapter.
in pathology. Okay,
Yes.
Doc. I don't have to know it now. Okay.
Yep. Okay, so let's speak about the synthesis of collagen. First of all, you need to know which cell it is. Because I know so many people who memorize the whole process, and the question is always like, which cell will be producing it, and they stuck. So it's the fibroblast. This is a fibroblast, okay?
Okay, Doc.
Okay, so this fibroblast, from the nucleus, we will have a collagen mRNA. It has a cap, it has a tail. The cap will be recognized by the rough endoplasmatic reticulum ribosome, and then they will initiate the process of translation. Yes?
Yeah, Doc.
So during the process of translation, the first type of collagen we make, it's called pre-pro collagen. So they write translation of collagen A chain, which is called pre-pro collagen, and it's usually a glycine XY. What is the XY? XY mean either proline or lysine. So if you get a question that tells you which of the following is the most common amino acid in the collagen chain, is it proline, is it glycine, or is it cysteine?
I would say glycine, Doc.
Glycine, because every other third amino acid is actually glycine. Yes? So
Yeah, Doc.
collagen is 1/3 glycine. Glycine content of collagen is less variable than the other two. Yes?
Yeah, Doc.
Hydroxyproline, however, is used for lab quantification of the collagen. Yeah, it's like just a test that we can do for quantifying the collagen. However, the most dominant amino acid in the collagen chain is glycine. Yeah?
Yeah, Doc.
Okay, so you can see here a pro alpha chain backbone. So there is a glycine and X and Y. So the X and Y are either proline or lysine. So hydroxylation of a specific proline and lysine residue, and it requires vitamin C. So the hydroxylation process requires vitamin C. I cannot emphasize enough how important is this concept. I mean that the hydroxylation process and the hydroxylation of a proline and lysine residue, they will ask you about it. They will tell you about it, and they will be like, oh, which vitamin does it require? The answer will be ascorbic acid. So again,
Anywhere else we see vitamin C, Doc?
in the
In
biochemistry,
such reactions? Yeah.
we will study them in the biochemistry chapter in the nutrition.
Okay. So
Yep.
Chelation. Okay.
Yep. So it requires vitamin C deficiency. It requires vitamin C, and in cases of deficiency, we will not have formation of normal collagen. Correct?
Okay.
So we get something.
So it is
Mhm.
this only for proline and lysine residues, though, not for glycine, right?
Yep. Yep.
Okay.
So we only
The major
hydroxylate
one
the
is not
proline
involved.
and lysine.
Only the. Okay.
Yep. So what is hydroxylation? It's the addition of an O and an H. Yeah? OH.
Doc, your voice is breaking a lot, though.
Let me try to recall you.
Okay.
Hey man,
Hey, Doc.
is it better?
Yeah, Doc. Better, Doc.
I reset the router. You know, man, in life you cannot get everything. You can get a beautiful city with a lot of holy sites with a bad internet. Yep.
Yeah, Doc.
So let's speak about the lysine and the proline. So hydroxylation of the lysine and the proline does require vitamin C. So as you can see here on the left, you can see the hydroxyl group is being added into the pre-bro collagen, correct?
Mm,
So pre-bro collagen was
yep.
the initial thing made from the chain, yes?
Yeah, Doc.
And then we hydroxylate it as a post-translational modification, correct?
Yeah, Doc.
Okay. So now it's hydroxylated. If I did not hydroxylate it because I don't have vitamin C, I will get a disease called scurvy. What is a scurvy? It's absence of collagen. If you don't have this collagen, what will be the problem? The gingiva will not be developing normally, okay?
Mm-hmm.
If gingiva is not going to be developed normally, they will start to bleed, yes?
Yes, Doc.
Yep. Okay, so what is
Doc, how do we differentiate this with this thing, Doc? With phentoin, calcium channel blockers, we have gingival hyperplasia, right?
I was just going to tell you about this.
Okay.
So how can you differentiate between scurvy and cases of using, for example, calcium channel blocker leading to hyperplasia? Well, the answer is always the history. If you have a patient who's always at home and he has this diet of soda and hot dog, okay? Soda and hot dog, they call it in the USMLE.
Okay.
You need to think about scurvy. Why? Because he's consuming zero leafy vegetables, yeah?
Okay.
Yep, so he will be in a really bad state. Okay, so hydroxylation of a specific proline and lysine residue require vitamin C, and deficiency of this process can lead to scurvy. Like
Also in UL, I saw a patient who only has tea toast, like a toast with a tea.
Yeah, toast and tea as well. Hot dog and soda.
Old.
Yep.
Okay.
Yep. Probably, let me guess, his wife died or her husband died. Yeah?
Yeah, Doc, something. An old lady having tea toast every day.
Yep. Okay, so glycosylation of a pro alpha chain hydroxylysine residue and formation of a pro collagen via hydrogen and disulfide bound. So let's try to understand what happens now. Okay, so we done the first step, we done the next step, now we have the third step, which is glycosylation, adding sugar. So it's glycosylation of a pro alpha chain hydroxylysine residue and formation of a pro collagen via hydrogen and disulfide bound. So I know this is a lot of words, but what I want you to know that we do add the sugar into this thing, but where do we add the sugar? Remember when we spoke about hydroxylation, I told you we hydroxylate the proline and lysine, but we do not hydroxylate the glycine, correct?
Yeah, Doc.
Now we glycosylate the hydroxylysine, but not the hydroxyproline, okay?
Only lysine, no proline.
Yeah, but not lysine, hydroxylysine, okay?
Okay, Doc.
All makes sense?
Hydroxylysine. Actually, not very clear, but I don't know what are you trying to say, Doc.
So, I was telling that like the process of hydroxylation happens at both lysine and the proline, correct?
Go ahead. Yeah.
But the process of glycosylation, which is adding a sugar, is only happening at the hydroxylysine, not at the hydroxyproline. So one less. Yeah.
So in second step we have hydroxylysine and hydroxyproline. In third step, it's only glycosylation is only at a hydroxylysine.
That's it.
Not hydroxyproline.
Yep. Okay, good. So they form hydrogen bound to bind to the hydroxy lysine. Okay?
Okay, doctor.
Okay. So if this thing does not happen, you get osteogenesis imperfecta. You know what is osteogenesis imperfecta?
Brittle bones, right, doc? Very.
Yep. So the patient is at high risk if he does not do this process, the glycosylation process, correctly. He's at risk of osteogenesis imperfecta. Okay. So far we have these processes: formation of the pre-bro collagen, formation of the hydroxylated form of collagen, formation of the glycosylated form of collagen. They want you to know that all of this process is occurring inside of the rough endoplasmatic reticulum. Okay?
Got it.
So everything until you make the procollagen, from pre-procollagen until you make the procollagen, all of this process is occurring inside of the endoplasmatic reticulum. They want you to know that. So then we have something called exocytosis. Exocytosis of a procollagen into the extracellular space. So we have the procollagen going to the extracellular space, going outside basically. Yeah?
Yeah, doc.
Okay.
By clathrin, right, doc? Lysosomes?
Yep, absolutely. So now it's
Golgi apparatus to lysosomes or Golgi apparatus to the cell membrane.
So now it's kicked outside. Now we need to do some more modification. So we did the chemical and metabolic modification, which was the glycosylation, hydroxylation, all of this stuff. Now it's time for the trimming. Yeah? So what do you trim?
We trimmed N and T terminals.
Yeah. So there are
T and N terminals, yeah.
They write exocytosis of a procollagen into the extracellular space, proteolytic processing, cleaving of the disulfide rich terminal region of procollagen, which is insoluble of a tropocollagen. Let me explain this point. So as you said, like we do the trimming. How do we do it? There is something called a disulfide rich terminals. What are these? The N terminal and the C terminal. Okay? So you just cut them out. And what did you cut out? This is the procollagen, yeah? So you cut out this section and you convert the procollagen into something called tropocollagen. Okay, so the procollagen is converted to tropocollagen. So far we have converted the precollagen with the glycosylation, hydroxylation into procollagen. And the procollagen, after the cutting, it was converted into a tropocollagen, correct?
Yeah, doc.
So the tropocollagen then, we take it and we start to do a procedure called cross-linking. Okay? This cross-linking is going to cross-link many collagens at the same time. So they write cross-linking, reinforcement of many staggered tropomyosin molecule by covalent lysine hydroxylysine cross-linkage, which is happening with the help with lysyl oxidase. So to connect these together, to make this bound, yeah, you need lysyl oxidase, which is an enzyme really important with a relationship to one disease, lysyl oxidase. And this enzyme requires copper to work, okay? Copper to work. So do you remember which two patients have a problem with copper? Either too much copper or too less copper?
Too much is Wilson's, right, doc?
Too less?
Too less is Menkes.
Yes. Okay, so cross-linking of collagen increases with age, and the problems with the cross-linking lead to Menkes disease. Okay, so if I'm going to cross-link more collagen, I'm going to start developing wrinkles, correct? Yep, that's why people get wrinkles, because of the cross-linking. Okay, what about
too much cross-linking, doc, or too less?
Too much.
Okay.
Yep.
Okay.
So you are not making more collagen. You are cross-linking it much more often. Yeah?
Yeah.
So the next one is osteogenesis imperfecta. It's in perfect.
Okay, let's take a 10-minute break, though.
Okay.
I just want to stretch my back.
No problemo. See you in 10.